Hey, thanks everybody for coming. Those that are here, and yeah, I need to move. And those that are right now listening to everybody by Zoom. So it's a real privilege. It's really a pleasure to introduce Quinton Smith. So I will be extremely brief in terms of introduction. So he has a lot to share. He received his master's in chemical engineering, and then went to Hopkins for his PhD in chemical and biological engineering. He was at the Sandia National Laboratories in Albuquerque, and then he went to Johns Hopkins for his PhD, and then he went to MIT for his postdoc as an HHMI Hannah Gray Fellow. Specifically in the last part in MIT, he was working developing biliary tissue engineering. So he received tons of awards. I was just chatting with David Gonzalez. Important, the Keystone Symposia Fellow, which David is also a fellow. And also he's currently funded by the Hannah Gray Fellowship from Howard Hughes Institute. He received many awards. He has been giving tons of lectures. It's a really pleasure to have him here. We have been collaborating with Quinton for quite some time. I'm not so sure he will be mentioning that right now, but he has this wonderful blend of engineering, biology, and right now focusing on stem cell fate decisions based on the microenvironment. That fits very nicely into this new area of going from the cells to tissues to organs, in our particular case, focusing on cancer. So it's really at the intersection of biology and engineering what makes him so special. He comes from very just 1 hour away from here, and UC Davis, UC Irvine, and he promised to come as often as there's an opportunity. Okay, so Quinton, without further ado, we look forward to your presentation.
Okay, can you guys hear me okay? Thank you so much for the kind invitation. It's been really rewarding to be here at UCSD. It was only like an hour ride here, but I definitely have to come here more often. So I'm really excited to talk a little bit about my PhD work, postdoc work, and kind of integrating into my independent research laboratory at the University of California, Irvine. And it's really predicated on like this image back here. So this is actually a micropattern array of stem cells that have been differentiated towards vascular lineages. You can see some micropatterns are all red, which will represent the vasculature. And some of those micropatterns are actually green, which will represent the pericytes. So I've been always curious about what actually dictates how stem cells actually make their fate decisions. I'll talk a little bit about how we can use engineering approaches to kind of tune their fate specification. So my research is motivated by a pretty grim statistic. So in the United States alone, over 100,000 people are in need of a life-saving surgery. So in the majority of cases, patients are actually in need of a kidney. But when it comes to the liver, which is the largest internal organ in your body, there's actually no treatment. There's no FDA-approved treatments, and the only thing that we can actually do is an orthotopic liver transplant. So, there are biomedical engineers that can create like pacemakers and dialysis machines that can actually serve as a bridge to transplantation. But the liver does so many different things. It's so many different cells. So I'm really interested in actually utilizing stem cells, which are a unique population of cells that have the ability to indefinitely self-renew and can be specified to any cell type in the body, to actually use these as a potential cellular therapeutic. So, in the case of the liver, with 200 billion hepatocytes, we actually only need about 10% of that to actually take on some of the metabolic features. But we definitely have a challenge in kind of getting our stem cell derivatives to actually take on functionality of adult tissues. So, while stem cells are, a great tool for understanding developmental biology and kind of getting to the point of understanding or getting to the point where we can actually develop cell-based therapies, there are still a lot of limitations. We really actually have a poor insight into basic human developmental events. So we're trying to come up with better ways to build models of early development. And once we kind of have these protocols to actually get the cell choices that we want, there's really poor differentiation efficiency in many cases, and we can't actually get them to reproducibly expand. And finally, we need to develop tools to actually ensure that we can actually evaluate their function and fate. So as Dr. Gutkind mentioned, I'm a chemical engineer, so I'm really excited about implementing engineering approaches to actually study how we can actually get the stem cells to mature in the way that we like. So, some of these include using microfabrication technologies such as micropatterning, or using different biomaterials, for example, as a way to control the niche or the microenvironment in which stem cells reside. So I really think tissue engineering as a whole really hinges upon a vascular bed. This is going to be the conduit that's responsible for the diffusion of oxygen, removal of waste and nutrients. So we actually have this process of de novo blood vessel formation called vasculogenesis. And these blood vessels continue to mature in a process called angiogenesis, where they continue to remodel and sprout. And again, as an engineer, there's a lot of different aspects of the vascular biology which we could perturb. So you can imagine, for example, you have high blood flow within your arteries that are highly oxygenated. But you can actually see, let's say in a case of atherosclerosis, you have like hardening or stiffening of the arteries. These vessels might be occluded, which is going to kind of restrict blood flow. So we can actually perturb these things using different in vitro tools. So how do we actually get stem cells to differentiate into vascular cells? So there's many different types of approaches. We typically employ directed differentiation approaches. But I would say there's been like a resurgence of these kind of embryo body techniques that I'll talk to you guys a little bit later today. But typically we can take these stem cells and we can actually coax them to go under different lineages. So I'll discuss, a basic protocol for inducing early vascular specification. And this is a 12-day protocol where we can take our stem cells and we can actually undergo a mesoderm induction, which is the germ layer that's responsible for cardiovascular lineages. And then we can continue to mature them into early vascular cells. So I started this is like an older technique of maintaining stem cells. Those large colonies are the pluripotent cells that can give rise to different adult tissues, and those cells that are kind of bordering our mouse embryonic fibroblasts that are actually secrete factors to maintain these cells in an embryonic state. And we can add differentiation cues to kind of push them to these vascular populations. So we can see after 12 days of differentiation in vitro, we can get a little bit of our population of endothelial cells, which are shown in red with this V-cadherin staining, and we can get some green cells that are going to represent the pericytes. So I want to tell you 3 different stories of how I envision or how I perturb the microenvironment. We can use micropatterning tools as a way to control stem cell colony size, and we can restrict the ability for these cells to interact. I'll discuss a little bit about substrate mechanics and then look into the roles of microfluidics and other techniques to control the architecture and allow us to impose different physiologically relevant forces on the tissues. So with micropatterning, we can really just look at basic questions such as like the loss of pluripotency. So we can actually take a colony of pure pluripotent stem cells, and we can put them under different microenvironmental cues. So we can create these micropattern arrays, we can control their geometry, we can control their size, and we can look at how does the role of stem cell colony size impact their differentiation fate trajectory. And we can begin to model it with using different types of engineering technologies. So here's an example. Again, we have this nice pure population of TRA181 pluripotent cells, and we can see this through flow cytometry. And then we can place these cells on micropattern domains, and we can steadily look at the loss of pluripotency, evidenced by this loss of green marker expression over the course of 5 days. So we can put these cells on micropatterns, we can give them cues to differentiate, and we can just simply look at how does the initial conditions of this differentiating environment really influence their lineage trajectory. So to acquire this data, we can use image processing algorithms. So here's an example of 80 micron diameter micropatterns. And we can use a really quick MATLAB program where we can identify each of those individual micropatterns as a pattern registry. And then we can begin to do local calculations. So we can look at the number of cells that are present on each of those micropattern domains, and then we can co-localize them with the TRA181 expression. So we can do like miniature analyses on each of those individual micropatterns and build up a large data set, again, to look at how do those initial conditions dictate stem cell fate. So here's just an example of the fidelity of the program. We can kind of see this really nice localization of these pluripotent populations. We can begin to do quick annotation. And I won't get too deep into this, but we initially showed, and this is in collaboration with Sean Sun, he's in mechanical engineering department of Johns Hopkins, that depending on the pattern size, the stem cells would actually undergo a different differentiation kinetics. So we can see here on day 1 under initial cell seeding conditions that we actually see like this kind of bimodal distribution of cells, whereas on the larger micropatterns from 225 to 500 microns, we see more of this unimodal distribution. What's really important is we're using serum and other factors to push these cells to differentiate. And here, looking at stem cell fraction, we find that on the larger micropatterns, we see a steady shift where the majority of the micropatterns are no longer occupied by pluripotent stem cells. But we still see on these smaller micropattern domains that we actually do have like this resident population of stem cells that are present. And this is actually kind of interesting because as we differentiate cells in lab, we know that cell seating density is a very important factor. If you see too high, these cells are kind of restricted in their differentiation potential. So this was a way for us to really model and analyze early differentiation kinetics. So as I mentioned, there are different ways or different engineering technologies as a way for us to understand early developmental events. So here's a cross-section of a gastrulating embryo. This will set down the body axis plan of a developing tissue. I mentioned the mesoderm as my lineage of choice. I'm also interested in the endoderm, which will give rise to liver derivatives. But we're actually seeing now this explosion of technologies as a way to coax stem cells to self-organize as they do during development. So we can use these micropattern arrays or 3D culture as a way to get these polarized tissues that represent the polarity and the spatial organization of human development. So I was really interested in applying these kind of foundational knowledge to really studying the self-organization of stem cells on different micropattern domains. So here's an example. We can take a pluripotent colony of cells. We can put them on these micropattern substrates. We can allow them to explore their microenvironment. So you can see that these cells are initially kind of randomly orienting, and they continue to proliferate and grow, and they begin to be interfaced with their boundaries. And we can do this for many different types of patterns. And we found something very interesting. We actually found this enrichment of BRCA positive cells, which is going to be this mesodermal precursor that was all enriched at the periphery of these micropattern domains. So here's just an example of some of the different micropatterns that we can actually develop, and we actually see this enrichment or this annulus of mesodermal precursors that are present at the edges of the micropatterns. And there's a lot of interesting work here in terms of receptor availability at the edges of the micropatterns or size dependency. So we can do a lot of perturbations, again, to understand how microenvironmental cues really drive stem cell fate. So we looked and we found that this enrichment of this BRCA annulus was coincided with an increase of this phosphorylated myosin light chain, which is going to be an indicator of cytoskeletal tension. And we found that if we kind of disrupt tension using small molecules, we can actually ablate this ability or the self-organizing parameters. And again, you know, as an engineer and working with some computational scientists, which I think is a really great aspect of this type of work, is that we can, again, use this image processing across many different micropattern geometries, and we can calculate the contribution of tension, cell seating density, in terms of this ability to actually drive these BRCA positive cells. And we worked with some collaborators to actually develop machine learning algorithms where we can take density or tension that's imposed on the geometries and predict the ability for these cells to undergo mesodermal specification. And what was really interesting and nice about this initial work is that we found that we can maintain or restrict their differentiation potential on these micropattern domains. And we found that just temporary disruption of tension during mesoderm specification kind of led to profound downstream consequences. So we see in our control conditions where we continue to differentiate the cells over the course of 2 weeks, we still see this annulus in the spatially restricted vascular specification. So we see these red cells, these endothelial cells that continue to differentiate. But we find, again, if we just temporarily disrupt tension early on, we lose the ability for these cells to mature into endothelial cells. So in this first work, we really showed that we can use micropatterns as a way to control stem cell colony size. We can use this to understand differentiation kinetics, and then we can actually impose spatial geometries along differentiation, understand how cytoskeletal cues impacts cell specification. So while there's these physical cues that are like really important in development, as we kind of show in these micropatterns, a lot of people will use chemical modulation to drive different gene expressions. So, you know, one important parameter in mesoderm specification is canonical Wnt signaling. So in a Wnt off kind of complex, you actually see beta-catenin being destructed. You kind of have this APC axon formation or this complex that's present. But we can actually use different approaches. We can inhibit the glycogen synthase kinase to actually disassociate beta-catenin, where it can activate or have a Wnt on state. So, you know, there's these different approaches that have been utilized where we can actually see the shuttling of beta-catenin kind of junctionally to this cytoplasmic or nuclear expression of beta-catenin, indicating a Wnt signaling on. So we can use these molecules such as CHRR as a way to induce Wnt signaling, so it acts as an antagonist. So people have used these approaches to induce vascular cells. So this is work from Sean Palachek's group, where they're able to induce endothelial progenitor cells over the course of 5 days. And they use the CHRR as a mechanism to induce mesoderm specification. But they find along this endothelial differentiation strategy that they still kind of lack this mature marker expression. So there's a lack of von Willebrand factor, which is really important in blood clotting. So, in addition to these soluble cues that are being used to induce differentiation, we can actually use mechanical cues again. So this is actually taken from, Adam Engler here at UCSD. He's done a lot of this work. A lot of the foundation actually stems from Christopher Chen at Boston University, where you can actually take mesenchymal stem cells that have the capacity to turn into bone or fat. And depending on the context in which they're differentiating, you can actually push them to go to either lineage. So again, you can use micropatterns, you can change the degree of cell spreading, you can make a stiffer extracellular matrix environment. You can see that these mesenchymal stem cells that are grown under a stiff microenvironment undergo or have this propensity to undergo an osteoblast differentiation, whereas stem cells that are cultured on a softer matrix will actually undergo adipocyte differentiation. And work from Cyril Dupont and Stefano Piccolo's group were able to identify this YAP-TAZ co-transcriptional activator as this kind of like mechanical rheostat. So this YAP is going to act as a signaling readout for differentiation kinetics. So we wanted to use something that we use in the lab all the time. This is PDMS. It's a viscoelastic material that we use for our micropatterning or for our microfluidic devices. And we can tune the stiffness. We wanted to look at the role of tuning the mechanical stiffness in terms of early mesodermal specification for these vascular lineages. So what we can do is we can actually make thin films of these PDMS substrates that represent biological tissues. So this is on the range of 3 kilopascals, which is about brain tissue. We have softer matrices that are on the order of 1.7 megapascals. And we can kind of compare this to stiff surfaces such as polystyrene plates, which is typically utilized in the lab. And we find that we actually can replicate this YAP localization dependent on the softer substrates. And we can actually see the degree of cell spreading is also influenced when we culture cells on these various microenvironments. So we were really interested in seeing if we prime the mesoderm specification on these softer substrates, if it would increase our endothelial specification. And we kind of took a page out of Chrissy Anseth's group. She's at CU Boulder. She's done a lot of different biomaterials, again, looking at mesenchymal stem cells, and has shown that there's been some fate memory dependent on culture conditions really primed by the mechanical environment. So we looked at mesoderm induction on these soft substrates. We compared to polystyrene and these PDMS substrates, and we were looking into or interested in the ability to differentiate these early vascular cells after 12 days. And we can monitor YAP expression. But importantly, we found that on these softer compliant substrates, we actually saw the shuttling of beta-catenin and more of this nuclear localization compared to our 3 gigapascal substrates. And when we looked at gene expression activity normalized to polystyrene plates, we actually found a large enrichment of mesoderm-inducing genes that were present on these compliant substrates. And then we continued to differentiate these cells towards early vascular cells using our continued differentiation protocol after 6 days of priming on these PDMS substrates. And we found in comparison to our 3 gigapascal, which is just polystyrene plates, that we're able to rapidly induce endothelial specification, really showing that the early priming on the softer substrates were able to increase the specification of endothelial cells. We were also able to achieve this in serum-free conditions. So we find again that genes that are implicated in mesoderm expression or overall have higher expression using these softer substrates. And again, we see the shuttling of beta-catenin in these softer compliant substrates during the differentiation to mesoderm. Looking at flow cytometry, we're able to see that cells that have been primed under these compliant substrates actually really had high levels of mature endothelial marker expression that was on order of human umbilical vein endothelial cells, really indicating that we can really push and drive more of a pure population of endothelial cells with this mesodermal priming. And importantly, we can look at other functional markers of endothelial cells, and we find while we can get this von Willebrand factor expression on the 3 gigapascal substrates, we can actually increase this von Willebrand expression on using these softer substrates throughout the differentiation, really indicating that these early fate decisions that are present can help with the maturation of some of the cells. So here in this work, I was able to demonstrate that we can use different materials as a way to coax stem cell fate and their differentiation efficiency. So I'm kind of going way fast. So in this next part of the talk, I just want to highlight different types of approaches to control the architecture of the microenvironment. So here's an example of a microfluidic device that we can create using those PDMS stamps. So we can use a process called photolithography as a way to create different features. And we can actually replica mold off this PDMS to create these little square channels. So these channels are an order of 250 microns tall and 250 microns wide. And we can seed these channels with endothelial cells and hook it up to like a peristaltic pump to simulate blood flow. And we can actually use this as a way to look at the role of shear stress in really driving maturation of our stem cell derivatives. So there's many different cues that are present or that are important in endothelial mechanosensation. This includes receptors such as the VCAD, PCAM, KDR complex that's been really pioneered work from Martin Schwartz's group. The glycocalyx has also been implicated in endothelial mechanosensing. It's like a sugary motif that's present on the outside layer of these cells. There's ion channels. I was really interested in primary cilia. It's this kind of microtubule-based organelle that sits outside of the cell and acts as this little sensor. So I was really interested to see if this primary cilia was really important in the ability for our stem cell derivatives to sense shear stress. So here's an example. This is actually from my lab notebook of some of the microfluidic devices that we can create. And we can see this hallmark phenotypic realignment of endothelial cells once they're put on these devices. So we see that endothelial cells that are cultured under static conditions adopt a cobblestone morphology, and they undergo this rapid realignment after exposure to shear, which we can analyze and quickly annotate with some of the image processing tools that I described before. So we looked at different mature and progenitor endothelial cell populations, and we were able to annotate and describe their ability to form primary cilia after serum starvation. So this microtubule assembly of these cilia projections are tightly in tune with their cell cycle. So we were able to serum starve these cells, and we were able to look at the degree of ciliation on these endothelial populations. We were also interested in looking at the shear responsiveness of these cells under different cytoskeletal conditions. So here we looked at our HUVECs, which are going to represent our mature cells, and we can add a drug called cytoclasin D, which will disrupt the stress fiber formation of these endothelial cells. And then we can introduce shear stress, and we actually find that these cells are still able to elongate and align. But we find if we disrupt microtubule organization with a drug called nicotazole, we find that while the cytoskeleton is still intact, we find that these cells are no longer able to align to shear stress. So we were really interested to look at what is the developmental context, or does primary cilia actually play a role in endothelial functionality? So this is beautiful work, not from my group, looking at zebrafish development. This is a FLIC1 or KDR positive mouse, where they're able to look at early progenitor blood vessels within zebrafish. And what they found, which is really interesting, is that you can actually identify primary cilia that's present on developing endothelial cells of the artery and the vein. So this is really beautiful work where you can actually see the cilia deflection in response to the viscous forces from the blood. So we can actually see here 24 hours post-fertilization along the endothelial layer, we actually see these primary cilia projections pointing up into the luminal space of the blood vessel. And then over time, we begin to see this primary cilia bend 28 hours to 24 or 26 to 28 hours post-fertilization. And upon this bending of the primary cilia, we actually see this increase of calcium. So it acts as this kind of like calcium handler. And what they showed, which I thought was very interesting, is that if you perturb different aspects of the flow environment of the developing endothelium, you actually get a disruption in some of the vascular maturation. And this is indicated in vascular loops. So for example, they can do a silent heart mutant where you actually don't have blood flow that's present. You can actually do this perturbation within the primary cilia itself. This is this PKD mutant. Or you can actually do a GATA1 mutant, which actually depletes blood So you still have flow throughout the heart, but the lack of blood actually doesn't lead to this viscous force. So it's not just the fluid velocity, it's actually the drag force that causes the cilia to bend. And they find that this led to less vascular maturation. So I was really interested in looking at cilia presentation in our stem cell-derived early vascular cell population. So we found, to much of my dismay, that our non-endothelial cells, so in green, we're looking at VCAD expression. So our non-endothelial cells were able to rapidly form these primary cilia projections. So we wanted to look at the capacity for these cells to align. So, we used our microfluidic devices, and we were really interested in getting these cell structures to align. And we found that in this mixed population, that under 5 dynes was just more venous shear stress, and 20 dynes, which represents arterial shear stress, that we found that our pericytes that had this primary cilia projections were able to phenotypically align kind of vertically, but we actually couldn't get this nice elongation of our endothelial cells. So we thought it was probably like a contamination of having both populations present. So we used magnetic activated cell sorting to isolate our endothelial cells. And you can see here, we have this nice VCAD positive population. They have eNOS, von Willebrand factor. Importantly, they don't demonstrate any pericyte markers. And again, we still found that our cells were not able to rapidly elongate and align to shear stress. And interestingly, we found very little of these primary cilia projections that we initially thought would be present within our endothelial populations. So we thought maybe it was our differentiation protocol that was impeding their ability to kind of functionally mature with these cilia projections. And we continue to look into other stem cell sources with using this protocol. So we found that we can actually get similar levels of VCAD, KDR, or other progenitor endothelial markers. And we found that our BC1 derived that did not have the primary cilia, and our new stem cell source actually didn't have a preference to arterial or venous specification. And we found using this protocol, again, that we were able to expand and define this population for at least 5 passages. And again, we found that our non-ciliated BC1 stem cell line, actually after serum starvation, were no longer or weren't able to actually form these cilia projections. But we found that our C12, using the similar differentiation protocol, or stem cell derived endothelial colony forming cells from a collaborator, were able to actually rapidly nucleate these microtubule projections. And after challenging these cells with shear stress, in this case, we looked into various levels and times with using our non-ciliated cells. So we did this for up to 96 hours. We found that our cells were not able to phenotypically align, but we found that our ciliated subpopulations of the stem cell derivatives were able to rapidly align and elongate in response to shear stress. In addition to looking at their ability to align to shear stress, we were able to use this microfluidic device as a way to look at calcium handling. So we were able to do a stepwise introduction of shear, and we were able to look at the uptake of calcium in response to these mechanical cues, and found that our non-ciliated BC1 stem cell derivative actually had this aberrant uptake of calcium, whereas our ciliated cells had this nice influx of calcium that was present. In addition to this lack of calcium handling, primary cilia has been implicated in kind of directional migration upon a wound stimuli. So we did these very simple scratch assays. And we found that, again, our non-ciliated cells were unable to properly close the wound, whereas our ciliated populations were able to rapidly migrate. And then when looking at their directionality, we actually found that our non-ciliated populations weren't able to orient themselves properly to the stimuli. So we looked into other molecules or factors that mediate microtubule assembly and found that histone deacetylase is responsible for the cilia dynamics. And by treating our cells with a drug called tubistatin, we were able to recover the ciliogenesis within our stem cell-derived ECs. So we actually found that we had this overactivity of this histone deacetylase implicated in microtubule stability, and we're able to recover some of the ciliation within our population. So after tubistatin treatment with our stem cell derivatives, we were able to actually get modest functional recovery, looking at our scratch assay that's present here. So our tubistatin populations were able to kind of recover their ability to directionally migrate. And we found that we were able to get a modest degree of realignment of our cells after tubistatin treatment. So with this work, we were able to demonstrate that we can use microfluidics as a way to really create functional microenvironments in which the stem cells reside in, as a way to look at their ability to functionally mature. And we find that stem cell origin might play a very impactful role in the ability for these cells to differentiate and have downstream functionality, and that this primary cilia might be kind of a progenitor marker or a way to assess the functionality of these endothelial precursor cells. So in addition to like the vascular tree, that's of course really important in sustaining tissue homeostasis, during my postdoc, I was really interested in this other type of tree. This is the biliary tree. And this is responsible for carrying out the bile acids that hepatocytes secrete. These are the main functioning cell types of the liver, into the small intestines where it aids in digestion. So I was really interested in understanding some of the ability for these cells to undergo this kind of morphogenic processes in the lab. And we're motivated by several rationales. This is work that was actually done by a postdoc at the time. She's now a professor at the University of Washington in Seattle, Kelly Stevens, where we were able to show that we can actually create these functional human-derived grafts for treating liver injury in mice. So we're able to functionalize cadaveric human hepatocytes using this aggregation technology where we can co-culture hepatocytes for fibroblasts to maintain their stability. And then we can create these endothelial cords using a micromolding technique. So we can create these vascular conduits that are there to act to support our hepatic cells. And she tested this in an animal injury model of something called tyrosinemia, which is a metabolic disorder that leads to liver failure. What's really interesting in this mice is that we can actually use this drug called NTBC that actually will kind of cycle the liver injury within this mouse model. So it acts as a kind of a block for this tyrosinemia background of the mouse to actually undergo cyclic injury. So we can actually induce different rounds of regeneration of the liver. So what she found was that when we place these liver grafts ectopically, if we underdo this NTBC cyclically to induce liver injury, that we can actually get these hepatic seeds, at what she called these hepatic seeds, to actually grow. And what was really interesting within this work was that she found a small population within these liver grafts were able to transdifferentiate into a biliary population, which is shown here. So we found again that we were harnessing the ability of the liver to regenerate to actually boost the engraftment and expansion of these human constructs made in the lab as a potential cell therapy for these animals that have liver disease. But given the necessity of these biliary tracts that begin to develop, I was interested in incorporating this biliary architecture in some of our tissue engineered grafts. So again, here's an example of the biliary tract that's present within the liver. So I was interested in using different types of tools to direct their self-assembly for their eventual integration with some of these bioengineered constructs. In addition, I wanted to use a model to actually understand what are the principles behind some of this tissue morphogenesis. So here's an example of a monogenic disease in the knot signaling pathway that's really important in cell-to-cell communication. And we find that one of the largest pediatric transplants for livers actually due to Alagille syndrome. So this is actually a monogenic disease that leads to bile duct paucity. So there's a lack of these biliary architectures that are present. So I wanted to see if we can kind of recapitulate this using an in vitro model system. So we were able to take human intrahepatic cholangiocytes. These are cells that are coming from the liver. They have a very distinct profile. And we were able to demonstrate different degrees of their functionality in the lab, including the uptake of alkaline phosphatase and their ability to undergo glutamyl transferase activity. And we found that we can actually use a blend of natural biomaterials, namely matrigel and collagen, to induce their self-assembly into a branching architecture. And we can do this under the administration of different biologically relevant growth factors, such as EGM. Interestingly, we found that if we use Notch inhibition that's been implicated in Alagille syndrome, that we can replicate this lack of branching morphogenesis. So we see that we can develop these functional branching-like structures that represent the biliary tract. And we find that this ability to form these branching architectures are really predicated on the microenvironment in which we grow these cells in, and kind of giving them the right growth factors to self-assemble into these structures. So again, to further our in vitro model system of using these primary intrahepatic cholangiocytes, we used CRISPR-Cas9 technology as a way to really ablate Notch receptor or Notch signaling within these cell populations. And we found again that under our ideal or optimized conditions to facilitate their self-assembly, that if there is disruption in our Notch tube, we can actually ablate their ability to undergo branching morphogenesis, which really takes on features of Alagille syndrome. While we're able to elicit their self-assembly using these natural biomaterial scaffolds, we were interested in using what we call these soft microfluidic devices. So we can actually create these instant perfusible lumen systems by using microfluidics and acupuncture needles. So we create these devices where we actually use these needles that can range in diameter to create these instant luminal structures. And then we can cast a hydrogel, whatever material that we want to use, around these needles. And then we can pull out these needles and then we can introduce cells there. So that gave us a way to create these hierarchical structures of the self-assembled branching structures with these larger luminal structures themselves. So in addition to using these primary intrahepatic cholangiocytes, we also looked into these assembly of rodent cholangiocytes. And again, we find that when we put these cells within a 3D matrix, they kind of self-assemble into these cystic structures. But we really wanted to find a way to really induce their branching architecture. So we looked into, again, soluble cues that are present from other cell types. In this case, we use fibroblasts. And by culturing these cells within this configuration, we find that cues that are indicated from multiple populations are really important in really driving branching features. And again, we can kind of use these engineering technologies as a way to create these instantly perfusible vessel-like structures in the group. Okay, finally, I'll do a quick overview of other techniques that I kind of did within my postdoc. So, it's really difficult for us to actually culture hepatocytes within the lab. And there's really a lack of understanding of really how does the liver regenerate. So the liver has this amazing capacity to regenerate. You can remove 2/3 of it and your liver will completely grow back. And there's really interesting studies where you can actually do these parasymbiotic studies where you can connect the blood vasculature of different animals, right? And you can injure one animal and you can actually get the liver to regrow in the other animal. So we know that these regenerative cues are actually present within the blood. But there's been a lot of debate within the field of where do these liver cells, or how do they actually get this capacity to regenerate? So really early on, there's just an idea that you have these kind of liver progenitor or stem cells that are present that are axin-2 enriched. These are cells that are sensitive to the Wnt signaling pathway, which has been implicated in a lot of the organoid technology. And the idea was like, oh, these axin-2 positive cells are the drivers of liver homeostasis and regeneration. And this idea that under chronic injury, you can actually get this transdifferentiated phenotype. So you can actually see, for example, if that patients that have cirrhosis or really far along progressed livers that have liver damage, those cells of the biliary tract will actually differentiate into hepatocytes. So there's a lot of this fate switching during extreme injury. But recent studies have actually shown that there's not like a progenitor cell type that's in the liver, and all the cells within the liver have this capacity to expand. So there's been really a not clear understanding of what drives hepatocyte growth and proliferation under injury. So we were really interested in really developing new in vitro culture methodologies as a way to support human hepatocytes, as a way to elucidate some of these mechanisms of liver regeneration. So again, we can use these micropatterning tools as a way to control architecture and drive functionality of different populations. So here's an example of freshly isolated human hepatocytes grown on these micropattern islands. So we can actually see some of the polyploidy of these hepatocytes on these micropattern domains. And we find if we just culture these hepatocytes in the lab, after a week, they actually completely de-differentiate and they lose their phenotype. So they need additional structural support. So we can actually induce these micropattern co-culture techniques. We can introduce almost like the stem cells that I mentioned very in the beginning of the talk, these feeder layer stromal cells. We can use these feeder layer stromal cells as a way to support human hepatocytes in vitro. And we can see one of the hallmark functionality of these hepatocytes are their ability to produce albumin. So we see under micropattern co-culture, with the incorporation of these support cells, they're able to continue to undergo their functionality. They don't lose their phenotype in the lab. But again, if they don't have this structural support, they rapidly degrade. They're no longer able to produce albumin. So I was really interested in seeing if we can adapt some of these techniques to actually really expand the utility of the primary hepatocytes using organoid technology. One of the downfalls of this technique is that we actually have to induce or use another cell population. So really important is really developing these in vitro model systems, for example, for drug toxicity. We know that 50% of clinical trials fail due to cardiac and liver toxicity. So developing in vitro models where we can actually test human liver cells to do these drug tests are really important. And by having pure populations of just hepatocytes will be an important barrier to cross. So I was really interested in trying to incorporate organoid technologies as a way to broaden the utility of these cadaveric hepatocytes that have to be stabilized with these complex micropattern or engineered methods. So really taking a page out of Hans Cleaver's books, we were able to optimize organoid media conditions where we can actually take single encapsulated hepatocytes within a 3D microenvironment. We can actually drive their growth as these organoids after 2 weeks. So we're able to look at different functionalities. So we can look at our micropattern cold culture. Here we're looking at 2 different primary cadaveric isolates of hepatocytes. So we can look at our micropattern cold culture where we have this arrangement, spatial arrangement of hepatocytes with fibroblasts, or we can look at our expansion media conditions that can really drive this organoid expansion within 3D. And we actually find that we're able to induce albumin expression or albumin production, as well as nitrogen handling using these types of expansion media conditions. Really interestingly is that we can actually look at their drug metabolizing activity. So this is a CYP3A4 activity assay. And we actually find, depending on the culture condition, whether they're in 2D expansion conditions or in just 3D basal media, that we have different degrees of drug metabolizing capacity. So we can look at this in a more refined manner. We can use rifampicin. It's an antimicrobial that's actually induces CYP3A4 activity. And we actually can find that from our screen using these different culture techniques, that we can actually stabilize these primary human hepatocytes without fibroblast support, and we can actually get them to undergo this rifampicin-induced CYP3A4 activity, which demonstrates that we can maintain the functionality of cadaveric hepatocytes without the addition of these stromal cells or another model, another cold culture. So here's an example again of CYP3A4 induction using rifampicin. We find that using just our 2D expansion conditions without this fibroblast population is able to sustain their functionality. But when we look at cells that are just cultured in 3D using basal media conditions, or in 3D in these expansion conditions, they're not able to maintain their CYP3A4 activity. So we find that actually we can actually get a boost of proliferation using these expansion media conditions. We can actually get a 3-fold increase in hepatocyte expansion over the course of 12 days. And we find that these cells are still competent in their ability or their sensitivity to different drugs. So for example, we can look at the toxicity of these expanding hepatocytes to different drugs such as acetaminophen. And finally, we can really characterize the populations of these cells using single cell sequencing. And we actually find that we get after 12 days of growth within these expansion media conditions, that we can actually get this rapid proliferation of these cells at day 2. So we can actually get a boost and increase their longevity in their functionality. And which is really nice, we actually see this increase in cell cycle projection. This is just a subset of the genes. But we see in day 2, during expansion, we're able to induce cell cycle progression, and they actually maintain their hepatic phenotype. So we see, from these Tisney representations or these UMAP representations, that we're able to maintain hepatic identity, but there is a subpopulation of biliary cells that actually arise during these expanding conditions. So hepatocytes are difficult to maintain in vitro. We need to support them with other cell types using kind of these fancy culture method systems. And if they do have the ability to expand, they still kind of have this biliary phenotype. So I'm really interested in really expanding some of these tools in my independent group. So as a new professor, I started last year, so actually April 2021. So it's been exciting, and I'm excited to share some of the work here. I'm really interested in, again, in integrating some of these engineering tools as a way to develop these functional tissues. So I have wonderful postdocs. I have 3 great PhD students that I'm glad, and we're embarking on these different projects together. So I mentioned it's a hurdle actually, using these primary human hepatocytes. You can't stabilize them in the lab. And even if we do have the ability to expand them using organoid technology, they still kind of have this dedifferentiated phenotype. So I'm really interested in trying to push functional hepatocytes for my PhDs in my group. And I kind of have some different ideas. One of them is that during liver development, the liver actually has a tightly in tuned crosstalk with the cardiac mesoderm. So heart development and liver development are really close to one another. And I think as we have different differentiation protocols, that we're missing some of the soluble cues that are present between this crosstalk that limits their downstream functionality. It's actually really interesting that like some of the stem cell derived hepatocytes, they can't metabolize alcohols. They're like fetal hepatocytes in the lab. So we actually can replicate that. But if we're looking to use these systems for drug testing, we really need to develop strategies to make them mature like their cadaveric counterparts. So I'm interested in thinking that we can induce some of these mature phenotypes by cross-talking some of the cardiac mesoderm cues with a developing liver bud as a way to derive functional liver tissues. So I have a wonderful postdoc, and we've been using kind of these 3D differentiation techniques, and we've been able to develop these vascular organoids. So after 25 days, we're able to create these self-assembled, really pure endothelial cells that are able to self-assemble as vascular networks in 3D. And she's able to actually integrate or develop also hepatic organoids. So we see here, we have hepatic nuclear factor 4 in green. We have this alpha fetal protein expression. It's not the best. There's still these progenitor hepatic markers, but we're able to generate these hepatic organoids in the group. And she's done some really cool work. We call these assembloids, where we're able to fuse our vascular organoids with our hepatic organoids to build, again, these stem cell derived functional tissues. And we're hopeful that these approaches can actually lead to the maturation of some of these stem cell derived tissues. Well, this is great. I think, this is a very crude method. We're taking spheroids that we've differentiated and we're trying to push them together and get them to integrate functionally. So I have a great grad student, Eric, who's been taking, this was his rotation project, where we're trying to use bioengineering approaches as a way to rapidly assemble some of these spheroids. So what he's doing is he's extruding spheroids, these stem cell derived spheroids, using a bioprinter. So we can create these different gels. We can actually extrude these spheroids in different orientations. And the goal is to be able to manufacture spheroids of vascular or hepatic origin into larger constructs where we can mediate their assembly. So I want to thank you guys again for giving me the opportunity to speak. This has been wonderful. And I just want to state that there's a whole open field of biology really looking at how do these mechanical cues or how do these microenvironments really drive stem cell fate. And I think this image is really amazing in the sense that these cells are able to survey their microenvironment, but their differentiation trajectory is very sensitive to these different cues. And as we look into the ability to form these self-organized structures in 2D, I'm really interested in engineering ways or looking at methodologies to pattern cells in 3D to kind of actually get more of a biomimetic system to study developmental biology. So with that, I want to thank people from my PhD lab, people from my postdoc lab, and my current group at UCI. So with that, I'd be happy to take any questions.
Thank you, Quinton. Usually we start taking questions from the audience, and then we go to the to the web. This time we'll do, it's not always the same, so we can do the other way around. So don't mind checking. Oh, okay, perfect. Adam, is he?
Hi. Yep. Can you hear me?
Yeah, perfect.
Excellent. I'm very sorry I couldn't be there in person.
So we cannot see you yet. Just 1 second. 1 second, Adam. Perfect. Now we see you.
Okay.
Awesome. Well, I'm really sorry I couldn't be there in person. But when life throws you lemons, make lemonade. So I'm glad that I could hear your talk via Zoom. And so typed one question in the chat. It was mostly about PDMS, and there's been this debate about how matrix proteins are tethered to their materials. I have very strong opinions, and we don't generally like to use PDMS as a result of sort of the chemistries that are involved with that. I was wondering if you could comment on the particular chemical linkages you were using, or were they just sort of fizzy-sorbed onto the surface?
That's a very good question. I think I read your Nature Materials paper specifically on this. So the idea is like, so we actually just use electrostatic interactions to mediate the adhesion. So we just coat these surfaces with collagen. But the idea is like these are viscoelastic materials, and that you can actually control where or the density of the adhesive ligands. So with these materials, you might have these clustering of, fibronectin binding motifs that cause changes within the material. So I think PDMS, to your point, is not the best model for a clean system of studying mechanotransduction. I do think maybe polyacrylamide gels or like a PEG hydrogel system where you can control directly the amount of RGD that you have is a better model system. But I still think, from this work that, by using these types of substrates, we can really get like a nice, elegant kind of response to these compliant substrates. And this reminds me of work from Jeff Milliman's group at WashU. He has a recent paper looking at pancreatic beta cell differentiation, and they use like a collagen gel height as a way to modulate the stiffness. Or, if you use natural biomaterials, if you increase the concentration, you're still increasing, the binding motifs there. So I think the best way to do this would be with a synthetic material, so you can actually decouple, ligand density, and the cross-linking that's present in these materials. So that's a very good question.
Yeah. No, and I say that not to downplay, anything that you see in PDMS, but more just to build awareness in the community that when one thinks that they've found something really profound, that confirming it on multiple types of materials, whether it's just different, hydrogel formulations, or even something different like a silicon rubber, as you see with PDMS, is really important. So I think all too often people just use one material. So I was not trying to single anything out, but just sort of build that awareness. I did have one other question that I was in the middle of typing when I got switched over to a panelist. So it disappeared, but I'll just ask it really fast if there's time. And that was with respect to what you were showing at the end from your lab with the endothelial-derived or iPSC-derived endothelial cells that you were sticking together in those assembloids. In other parts of your talk, you had some sort of sacrificial layer or like a rod that you were able to remove, and it created a patent channel that you could perfuse. And there I would call that, a rudimentary vascular network because it's patent. But here, I guess, and a lot of people do it in the field, they call them networks, but there's nothing actually moving or perfusing through the endothelial cells that you've got there. So when you describe them as a vascular network, what effect do you really think that you're seeing? Because to me, it doesn't seem like they're acting as true vessels, but more just as a paracrine depot. So I guess I wouldn't use the term vascular network.
Right. So that's a very good point. So again, I think this shows the integration of vascular cells. So we're using these vascular spheroids as a way to, show crosstalk between these different lineages. But to Adam's point, from Christopher Chin's work, Chris Hughes at UCI, we can actually develop these microvascular systems. So what I kind of showed with the biliary tree is that we have these perfusible ducts. So the goal would be to integrate some of these vascular cells. They do have lumen. We can confirm it through confocal, but integrate them in a perfusible system. So let's say we have in the center of these microfluidic devices some of our hepatic organoids, and then we have these patency within these perfusible structures. And we can actually get the blood vessels to self-assemble through the hepatic organoids, where we can actually flow dyes, for example, like dextrin, to show that these vessels not only can hold fluid, but they actually have strong barrier function. So I think to your question, that's the next step, right? So we can show that these stem cell derived cells can integrate with other tissues, which is not an easy feat. You take HUVECs, they don't integrate with every cell type. So I do think the stem cell derivatives are kind of privileged to interact with different mature tissues. And this can be evidence from Shaheen Rafi's recent paper with these reprogrammed ETV2 cells. So really interested in using our stem cell derivatives as like an analog of these cells that can integrate with other functional tissues, and then creating a perfusible or functional vessel device. Yes.
Thanks.
Sure. Thank you.
Thank you. Questions? Jing.
I'm very interested in mesoderm induction. So this is an elegant system, but I was a little surprised the data actually shows that the soft, I don't know, this tension you would say, actually promote mesoderm. I guess it depends on how much the tension, because earlier work have shown in Drosophila during this invagination, right before the mesoderm induction, the force is actually important for the induction of the transcription factors involved in the mesoderm, I guess, in the embryo. I'm just wondering whether there's a different type of force, because the invagination also causes, I guess, change of geometry of the cell in addition to the force, and how do you can you mimic this?
That's a really good question. So yeah, when I think of mesoderm induction, I think of a lot of EMT, a lot of increases in traction forces. So I think from like our in vitro system, we're adapting cells that have been primed on these really stiff substrates. So when we put them back to like a physiologically relevant system, we begin to, again, ask those basic science questions in terms of their sensitivity to the compliance of those environments. So again, also with the micropatterns, which are completely on completely stiff surfaces, we actually see this enrichment and higher tension at the edges of the micropatterns. And then we see on these compliant substrates, which are deemed soft, that they seem to undergo like an increased mesoderm expression. So I do think we need to be very careful in what we think about in terms of the in vitro context in which these cells are residing. Because I think these compliant substrates are maybe imposing different degrees of tension than these local boundary conditions that are from the micropattern domains. But in general, I do think like, maybe pushing these systems to 3D would be like a more physiologically relevant system to look at the roles of tension and mesoderm induction. But there's work from Valerie Reaver's group that shows very similar things where they can put stem cells on soft compliant substrates and they undergo efficient mesoderm specification. So I think the challenge is mapping some of the in vivo developmental systems to the in vitro systems. So yeah.
So what is fascinating, you can go from in vitro to in vivo. I mean, sorry, from stem cell to sort of mature function, and eventually also in vivo. So a quick questions. So I mean, fascinating. I did not know the endothelial cells have a lot of cilia. And so what would be the role of cilia, number one, in endothelial cells, if you can define it a bit? And the second is a role for sonic hedgehog signaling there, smooth on, etc.
Yeah, so I would say cilia has been implicated. So actually during development, the cilia actually becomes devoid. So under high shear stresses, the cilia is actually gone. But they actually find under like atherosclerosis or these regions of occluded arteries, the cilia comes back up. So I think it's acting as a signaling hub for relaying inflammatory stimuli from the local viscous forces that are present there. So I think it still acts as this mechanossensor, almost like cilia plays a role in like kind of polycystic kidney disease. There's this like really sensitive, sensor of osmolality and other different inflammatory cues. So I think, its true role in endothelial function, I don't think is fully elucidated. Like I'm not sure if it mediates like vascular permeability or growth, but we do know that it plays a very important role in its shear sensing sensitivity. And yeah, like as you mentioned, cilia has many different signaling pathways that it influences, including sonic.
So especially with smooth on systems, so would be very exciting. We can chat afterwards. For who? Last question.
Okay, so I wanted to ask a question about your grad work with micropatterned, the cultures on micropatterns. So we see kind of, you have a retention of pluripotency at the edges, and this is correlated with skeletal, cytoskeletal tension. It resembles kind of what we see at the invasive front in cancer. And I was wondering if you could comment, like, can you take this kind of from this very like defined system? Can you make any insights? And do you think that there's something at the cancer invasive front like mechanical forces that promote self-renewal that allow for further invasion?
Yeah, that's a very good question. I would think, this is a nice system. Again, I think this is a nice system again to control mechanics as well. So I think you can maybe induce an invasive front using different material stiffness as well, in addition to using micropattern geometry. So I just want to get at your question. So you're saying the receptor availability or the lack of ECM presentation is going to be a good model for like cancer motility or cancer metastatic potential?
At the invasive front, often we'll see, staining for factors that are associated with self-renewal. It may not be the same thing, like it may not be TRA1-81, but factor like we see things like YAP-SOX2 or some of the EMT factors at the invasive front. I don't know anything about the tension that's occurring there, but it's just like from a visual standpoint, it resembles the same thing. I know what's different is, I guess they don't have any adhesion on the other side of it.
But I think it's a good way to impose different tissue, like collective tissue stiffness. And you can see if there's self-emerging patterns based upon the local tension from the geometry. So if there's like a pool of different populations that might have more of a stem versus more mature progeny, maybe there can be a degree of self-sorting based upon tension using these approaches. So I think that would be like a cool project to put, maybe these cells in these different environments to see if these cells self-sort based upon the tension. So, yeah.
Perfect. So thanks everybody in the Zoom. Thanks everybody in the audience, and especially many thanks, Quinton. So it was amazing. So thank you. Thank you. Wonderful.